to introduce Professor Strew. Professor Strew has come all the way from Albion College to talk to the students today about his research and his career path. Professor Strew teaches biochemistry and other chemistry courses and does a lot of research with undergrad students. A little background that this talk is funded by ASBMB networking grant. And when I received that grant, I didn't even know Professor Strew, but fate had it, and I went to San Antonio for this national meeting by ASBMB, attended his talk, and his work is something related to what I do, so the first thing that came to my mind was collaboration. But then later on, a common friend introduced us, and that's when I thought, oh, this is the right fit. So here we are. Professor Strew will talk about his research, his career path. You have questions, anything you will feel free to ask. And there is some swag as well, which you can take while you leave at the end. And please feel free to ask questions related to the research, because he works with undergrad students, so that should be something relatable to you, and that should inspire you as well to work in our labs. And with no further delay, I will let Professor Strew talk to all of you, and let's give him a warm welcome.
All right. Thank you so much for the kind introduction. Thanks for inviting me here today. I'm really excited to be here. It's my first time on campus. We've got really nice facilities here. So like the professor said, I do drug design research, and we met in San Antonio. I gave a 12-minute talk in San Diego, which was really a 20-minute talk that I crammed into 12 minutes. And so I'm going to talk to you about a little bit more that I do with undergraduates. But I do want to talk, because I think probably the thing that is most interesting to you is sort of my career journey, or how I was thinking about these things at the time. And so I started at Albion College. I teach at Albion College, but I was a student at Albion College. When I decided to go to Albion College, I didn't make any it wasn't profound. I went to Albion College on the way back from Western Michigan University. There was a sign on the side of the freeway, and I was like, well, there's a college here, I should stop and look at it. And then people were kind of nice to me, and so I was like, hey, maybe I'll go here. And I was like, I'm gonna do architecture. And of course, Albion doesn't have architecture, right? But that didn't occur to me at the time. It was only after I got on campus where I was like, oh, I'm gonna do architecture. And they're like, no, you're not. And I was like, I need like a few $100 for books. I'm like, do you have any campus jobs? And they were like, no, but you can work in the lab of a professor. And they'll pay you a couple $100 for the semester to do that. And I thought, well, that sounds good. And they give me the list of professors. And so they gave me a list of professors. And I was like, I want to do rainforest biology, because it was like, well, maybe you'll get to go to the rainforest. And they're like, no, taken. And I was like, okay, I'm going to do genetics, right? Genetics, yeah. And of course, that's also popular. Taken. And so I was like, well, what do you have, right? And they were like, well, we have this organic chemistry slot. And I was like, now I hate organic chemistry. That's what I thought. That's what I thought at the time. I was like, I hate chemistry, in fact. And so I took this thing to my high school chemistry teacher. I'm like, this is the thing I'm going to go do. I'm going to make a few $100s. And she was like, I have no idea what that is talking about. And so I went and I did a thing, and I thought I was good. I thought I was going to hate it, but I'm like, hey, it beats, whatever, any job. I'll scrub dishes for $200 for the semester or whatever. And so that's what I did, but it turns out that I fell in love with it. And so I was sitting in my, I didn't know what I was going to do with it. I still wasn't convinced. I didn't see myself as a scientist. There weren't really scientists or chemists in my family. Everybody in my family was a teacher. So ironically, I'm now a teacher. I'm just a teacher that does it at the college level. So I never really thought about it. But my mom was like, you should be an accountant. You're sort of good at math. And I was like, but I don't want to be an accountant. So I'm still a junior, and I'm still taking chemistry courses, thinking I'm going to be a lawyer. And I'm sitting in the office of my undergraduate advisors, and I'm like, I hate everything. Everything in the world sounds like work, right? And I don't want to work for a living. And I was like, but your job. It's 3 o'clock on a Thursday, and you're talking to me about what I want to do with my life. That sounds awesome. I want to do that. And I was like, well, how do I do that? He's like, well, you got to go get a PhD. I'm like, well, how do I get a PhD? And he's like, well, you got to go to graduate school, right? I'm like, how do I go to graduate school? You got to take the GRE. And so one thing leads to another, and I went to graduate school. Now, I was set up pretty well to go to graduate school because I'd done some research, and I decided I was going to go to the University of Pennsylvania, because when I looked at all the graduate schools, the only one that I was really interested in at all was one professor at one school because he was making drugs, okay? And so that's where my talk begins, is I decided, I fell in love really early on with the idea that you could make people better, right, with pharmaceuticals, right? And so I set out to work with this chemistry professor. And he was making pharmaceuticals, but of course he encountered a challenge. He was a very young professor himself, pre-tenure, he's in his second year as a professor, and he was writing all these grants because he wanted to make drugs. But every time he would write a grant, the grant agencies would say, why should we fund this? Because you're competing with the pharmaceutical industry. So the pharmaceutical industry is large worldwide. It is $1.4 trillion in value. It has $80 billion in annual R&D investments, and it has 5 million worldwide industry employees. And at the time, the Meggers Lab at the University of Pennsylvania, it had state-of-the-art academic facilities. We had one of all the best equipment. They had about a $400,000 annual budget and 8 graduate students. And they're like, that's pretty good, but how are you going to compete? We also had Andre. Does anybody know Dr. Dre 4000? Andre was my neighbor. I worked next to him for 4 years in graduate school. So that was cool. Had a pretty cool neighbor. The pharmaceutical also has all these force multipliers. They have robots, right? So not only do they have 5 million employees, they also have robots that will do these things for them. So it's like, how can you possibly compete? And so in graduate school, we had to figure out, we had to do something that the pharmaceutical industry wouldn't do, right? And so over here on the right is a known drug. It's called staurosporin. It's a kinase inhibitor. Kinases are sort of the bankers of the cell. They take ATP, they phosphorylate those things, they turn pathways on and off, and so they're responsible for a lot of disease, most notably cancer. And we were like, well, let's make molecules that look like staurosporin, but instead of having a sugar, right, over here on the bottom, it'll have a metal complex. And the pharmaceutical industry wants to stay totally away from metals, except for the fact that they use metals all the time. So some of the most common cancer drugs are drugs like cisplatin and oxaliplatin, which are centered on platinum metal complexes. We also use gallium as MRI contrast agents. But the dogma at the time was that you never wanted to have a metal in a drug, despite the fact that some of the best-selling drugs of all time contain metals. And so the idea was that if you're going to just stick with carbon, you can make a variety of architectures. You can have things that are sp, which is sort of linear. You can have things that are sp2, which are flat. You can have things that are sp3, which are tetrahedral. You learn this in your introductory chemistry class. And those tetrahedrals can have 2 stereoisomers, so you have some variability available to that. But if you use a metal, you can make complexes that have 6 ligands. And if you can have 6 ligands, you can arrange those ligands in 30 different ways. And so you could make tons and tons of really cool drugs by rearranging the ligands on those metal complexes. And so that's what we set out to do. And we did that. I don't want to talk too much about the story because this is sort of what I did in graduate school. It's not what I do now. But we made a ton of metal complexes, and they were these beautiful colors, right? So most organic chemistry is pretty much clear or yellow. But when you start working with metals, you have these amazing colors. And we screened those for all different kinases. And we found different metal complexes inhibited different kinases. And that was really important because kinases are all the same. We have about 550 kinases in a human, and all of them bind ATP. So if you're going to make a drug that also binds in the same spot as ATP, you stand the chance of inhibiting many kinases, the ones you want and the ones you don't want. And so you generate off-target side effects. And so the idea was to pick out just one kinase. So we have MSK selective, GSK selective, and then BRAF selective. And so this is the one that I worked on most. And it inhibited a BRAF. BRAF is one of the kinases in the MAPK signaling pathway, which is one of the main signaling pathways in cell differentiation. And it would get this mutation. So this valine 600 would get mutated to a glutamic acid. So valine is, of course, a hydrophobic residue, and glutamic acid is a negatively charged residue. That negative charged residue would mimic a phosphorylation, because there's a phosphorylation event that happens on a tyrosine that's right next to this valine. And when it would get mutated, it would make a salt bridge with this lysine, which has a positive charge. So there was a negative charge here and a positive charge here. Nobody really knew how that worked. So we took our drug, we used our drug to solve the active crystal structure of BRAF. What does BRAF look like when it's on. And the reason that was important is because this mutant is the single most important point mutant in all of cancer, right? There is not one single point mutant that causes more cancer than BRAF V600E. And so based on that, I didn't do any of this. They took our crystal structure, they designed this drug, which is vemurafenib, was the very first melanoma drug to come out in many years. The prognoses for melanoma in the early 2000s were very poor. And so now with these drugs and checkpoint inhibitors, 5-year survival rates have gone from 5% for certain types of melanoma into the 90s. It's called vemurafenib because it was a V that was changed to an E, which was a mutant form of RAF. And then it's an inhib because it's a kinase drug. This caught lots of attention in the New York Times, USA Today, whatever, so on and so forth. And so that was kind of cool. It was like we made a difference in science doing a thing that the pharmaceutical industry wouldn't do. But around this time, I was out here doing I finished my PhD over here in Penn. But during my PhD advisor moved to Germany. So all my friends moved over here for 2 years, which was really cool because each year for several years, I would go to Germany to meet with my lab group funded by my PI, always at the end of September. What happens at the end of September? Yes, so I went to Oktoberfest twice because my PI moved to Germany. But when I was thinking about like what I wanted to do next, right? I had a family and I didn't really want to venture overseas. And so when I was thinking about a postdoc, I found sort of a lab at the University of Pennsylvania that was doing things very biological. Now the goal was always for me to become a professor. And I kind of always thought I might want to be a professor at a small place, right? And so to be a professor at a small place, you kind of got to be a jack of all trades. You have to do all the different things. And so I had learned how to make drugs. I learned organic chemistry, I learned inorganic chemistry, but I didn't really know how to test my own drugs. And so the idea was I needed to find a lab where I could learn how to do the biophysical tests that I needed to test the drugs. And so I found a lab, and they were doing the biophysical tests that I wanted. This lab, however, it was a hematology lab. I had no particular interest in hematology, but I went to work for the professor who discovered a protein called integrin. Integrin is a cell surface receptor, and the specific one that he discovered was called alpha-2b beta-3, which is the one that causes blood clotting in platelets, right? And so this is what heals wounds, but it's also what's responsible for heart attacks. And we were interested in all the biophysical testing of the proteins that bind to integrin alpha-2b beta-3, and so I spent like 3 years testing, working in biophysics, testing these things. So it was a really like sharp left-hand turn from organic, inorganic chemistry to biophysics. But now, I'm asking my postdoctoral advisor, I'm like, hey, I want to go find a job at a small school. And I was prepared to do that. I could do all of these different techniques. Now, at the time, he said he didn't know why I would want to work that hard. Because he thought that working at a small college was harder work than he had as an endowed professor at an Ivy League institution. And I thought, you're crazy. Working at a small school is going to be easier. Turns out he might have been right. Okay, and you can ask your professors about that as well. So when I went out to set out my own lab, right, I was like, okay, well, I want to make drugs, right? And then immediately people were like, but the pharmaceutical industry, right? Again, $1.4 trillion in value, $80 billion in annual R&D investments. But now instead of having state-of-the-art facilities, a $400,000 a year budget, I have shared space and equipment and somewhere between $1,200 and $8,500 in annual budget. Instead of 8 graduate students and a postdoc, I have somewhere between 3 and 24 18 to 22-year-olds, right? And so we have to somehow compete with this. And so that was the challenge that I originally started. And so I started thinking about the ways in which we compete. And I'm going to mostly talk about pharma's waste bin, okay? But we've been thinking about other ways that we can sort of be competitive with, or at least contribute to the to drug design. And so this is the island of misfit toys. This is more from my generation, right? But these are the toys that were discarded, right? And so they have sort of found a new life on their own island of misfit toys. And so that's what I think about when I think about the research that we do. So when a pharmaceutical company is making drugs, they might make 10,000 compounds. Actually, they're making like 10 million compounds. But for every 10,000 compounds, one of those compounds will make it to be a a drug that gets FDA approved. And so the attrition rate is huge. 9,999 of every 10,000 compounds gets scrapped. Those are the misfit toys, okay? And so what we tend to do is and the reason that we're dumpster diving at pharma is really what we're doing. And so the reason that those drugs fail is because it doesn't do the thing they want it to do. We're not interested in those drugs. We want drugs that do the thing that you want it to do, but they also have undesired or unintended side effects. And typically those undesired or unintended side effects are because the drug does a thing in your body that you didn't design it to do, right? So you made a drug that was supposed to be a kinase inhibitor, but it inhibits a phosphatase as well, right? And so you get some unintended consequence. And can we take those drugs that pharmaceutical companies have abandoned, that we know work, they just do a thing we didn't want them to do, and can we make them better, okay? And so the idea at the time was, hey, can we turn these drugs on? There is this entire field of science which is called photodynamic therapy, where you can turn drugs on with light. These cancer drugs already exist. And the idea behind that is that you can administer the drug systemically. So we've taken this green inactive drug, we've administered it systemically, and then we shine a light on a specific place. So in this case, we're shining a light on a tumor that activates the drug, and the drug does its thing. And these are really, most photodynamic therapy were cancer drugs. And the reason is because it was really easy to activate a drug that would just blow everything up, right? So you shine light on a thing and they would make free radicals and just explode everything, right? And so that's what you want to do in a tumor, right? But that's not exactly what you want to do if you want to inhibit other things for other diseases. And so it's all right, can we make our own version of photodynamic therapies? Well, we were inspired by a thing that you might have heard about in biochemistry, right? So so this is a cell, this is a rod cell that's in your eyes, and in those cells there are discs, and in those discs there is a protein, and in that protein, the protein is called opsin, and in that protein is a molecule called retinal. That retinal generally exists in the cis form, right? So this is that bent form. This bond is in the cis form. And when it gets hit with light, it changes into the trans form. That change from the cis to the trans form exerts pressure on the opsin protein, and that causes the opsin protein to signal downstream. And that's what our brain recognizes as vision or light, right? So we create a picture of it in our brains. And so that's what's happening, right? A molecule is isomerizing in response to light, and that's sending a signal to our brain. Okay, so there are certain problems with using carbon-carbon double bonds and isomerizing them. We'll see what that is in the next slide. And so there are double bonds that can isomerize much more easily than carbon-carbon double bonds. And these are called azo bonds. So this is an example of an azostilbene. An azostilbene is a nitrogen-nitrogen double bond in between two benzene rings. Here it is in the trans form. If you take and you shine short wavelength light on it, you can turn it into the cis form. And if you use longer wavelength light, you can turn it from the cis back to the trans. Now, you might know from organic chemistry, if anybody's had organic chemistry, that trans bonds are more stable than cis bonds, okay? And so this cis bond will slowly over time, in the absence of light, revert thermally back to the trans, okay? And so that adds another layer of difficulty to this. We have to turn it from the trans to the cis, or the cis to the trans, and we got to get it to stay there, okay? All right, you might know these azo compounds from such things as the M&Ms, okay? So Allura Red, or red dye number 40, is, it's in Code Red Mountain Dew, it's in the stripes on candy canes, it's in the shells on M&Ms, it's kind of everywhere, okay? And so we eat this stuff all the time, okay? So it's in biology. And so what's happening? So this is going to appeal to the physical chemists in the room. We got one slide here. Bear with me. So you've got a pi orbital. You got a pi bond, and there's 2 electrons in that pi bond. And those electrons, when we're changing this from trans to cis, we're taking those electrons, we're exciting one electron into a pi star orbital. And it is the distance between these two orbitals that relates to the amount of energy that we have to use. And so for a carbon-carbon bond, it takes like 200 nanometer light. Now 200 nanometer light is like really damaging to your skin because it does things like this. It excites electrons from carbon-carbon bonds. But in a nitrogen-nitrogen bond, those orbitals are much closer together, and so we can use lower energy light, right? So longer wavelength means lower energy. And so it takes about 380 nanometers. Now that's UV light, and what do we know about UV light for our skin? Yeah, it's not awesome, right? It's not good, right? And so that is how you go from the trans to the cis. What happens when you excite from a pi to a pi star, right, is that you get, this is how you would represent it, basically a broken bond. And then that electron can, you can rotate around that bond, that electron falls back down in energy, and you get a new bond formed. But now, because you're in the cis conformation, the amount of energy that is necessary to go from the pi to the pi star orbital, these orbitals are even closer now because you're in the cis form, is 470 nanometers, okay? And 470 nanometers, anybody know what color light that is? Yeah, it's kind of a blue-green, yeah. It's like a blue-green, yeah. And so, but that light is not problematic, right? So we can go from the cis to the trans using blue-green light. This is all, of course, defined by this equation. And the longer the wavelength of light, the further that it penetrates through tissue. That's also another thing that you want to remember. So when I was a kid, you could take a flashlight, you could put it under your hand, right? And what did it kind of look like? What did you see coming through? What color? Red, right? Now when you're a kid, you think that the reason it looks red is because there's like blood inside of us, right? Or something. I mean, am I the only one who thought that? No? Okay. It turns out that the red wavelengths are the only ones that will pass through our tissue. The other ones are absorbed, right? And so what we see is red, okay? All right, so we can take advantage of this, okay? So the trans version of these azo compounds is super flat, and we know that from like organic chemistry or general chemistry. They're all sp2 atoms. sp2 atoms are flat. But because there's a steric repulsion of the hydrogen here and the hydrogen there, in the cis form, which is why the cis is less stable, these molecules are bent. So they have really, really different structures. And so we can take advantage of that in this way. So sometimes in biochemistry, we'll teach the analogy. I don't want to call it the model because we've replaced that model with other better models. But the analogy, which is that drugs bind to their receptors like keys fit in a lock. They have to have a similar shape. They have to have a similar electronegativity. And so if we design these drugs just right, we can design them so either the trans or the cis will fit in the receptor, right? In this case, the receptor binds to the trans form is inhibited. And so we can create a drug that we can activate, right? Because that's what drugs do. They fit in the receptors and stop the receptor from doing what they're supposed to be doing. We can activate by changing a cis to a trans and activate the drug, okay? All right, so when I set out to do this for the very first time, I was like, let's try resveratrol. Now resveratrol was in the news. Everybody was talking about resveratrol at the time. Resveratrol is the compound that everybody was talking about because they compared the American diet with the French diet and also the Italian diet. And it turns out that like the French and the Italians don't eat less saturated fats than Americans, right? But they live an awful lot longer. And so they're like, couldn't possibly be diet or exercise, but they controlled for diet or exercise. And then they were like, well, it's got to be something else. And so they're like, wine. It must be wine that's doing it, right? Because that is what's different between the American and the French or the Italian diets. And so they zeroed in on red wine in particular. And it turns out that red wine, especially from volcanic soils, makes a series of phenolic compounds. One prominent one is resveratrol. They fed this resveratrol to mice or rats, I forget. And they did these studies where they would like have like a turning bar, right? And they'd see how long they could like stay on the bar. And then they would feed them different diets. And you were like trying to get a high fat diet. They would go to McDonald's, not kidding, they would go to McDonald's and they would get French fries and they would feed them. And so they did 3 sets of mice. They did mice that had a high fat diet and resveratrol, a high fat diet and not resveratrol, and a low fat diet. And they found that the mice who had a high-fat diet and no resveratrol, they did not stay on the bar very long. They did not live very long. They fed them resveratrol, and they lived exactly the same length of time as the low-fat diet mice. They were at least as nimble as the low-fat diet mice. although they did have higher body mass indexes. And so the thought was like, this is the compound that you're going to live forever. And I was like, well, I would like to be able to turn that on, right? And so I was like, well, it has a double bond, and let's put an azo in there. And so we did that, at the time there was some debate about how resveratrol was working. Is it binding to an enzyme called SIR2-1, or is it binding to an enzyme called quinone reductase 2? It turns out it's binding to both. But which one is the live-long activity, the longevity activity due to? So we teamed with some folks at Purdue University, and we crystallized our azo compound in an enzyme called quinone reductase 2, which is one of the enzymes they thought might be causing this activity. It turns out, so this is resveratrol, and this is azo resveratrol. It binds in exactly the same place. It inhibits exactly the same way. But along the way, it was discovered that, well, resveratrol does seem to work. We cannot attribute its activity just to resveratrol, because the amount of wine that you would have to drink, right, in order to get enough resveratrol works, but in order to get enough resveratrol, you need something like 20 gallons a day, which is approximately a bathtub, and the alcohol and other things would kill you far before the resveratrol would kick in. And the reason for that is because resveratrol has all these polyphenolic compounds. They get sulfonated and glycosylated, which makes them really soluble, and then they're excreted through the kidneys. And so we thought, well, let's make one that we wouldn't have to drink a bathtub of wine for. And so we made all these derivatives that lacked the polyphenols. So we did some medicinal chemistry. We made a variety of these different compounds. Like this compound can't be sulfonated or glucuronidated, and it has a similar activity to resveratrol. And so we thought, we've got the ticket, okay? And I want to turn these things on. So we had a good drug, right? But I wanted to turn them on and off. And so I need to talk to you very briefly about what we're looking for when we're turning these things on or off. So when you take a mixture of these compounds, you take like a UV vis spectrum. You may be taking a UV vis spectrum in general chemistry or something like that, where you shine light on and you see how much light the compound absorbs at different wavelengths. These compounds always absorb light at two different sort of wavelengths in the near UV to the visible region. There's usually a giant peak here, about 365, 380, and that is the light that is absorbed when this bond gets turned from a trans to a cis. There's also tends to be a smaller peak here in sort of the 470 range, where that is the light that is absorbed when you're turning from the cis to the trans. And we have names for these, right? These are both pi to pi star bands, but that's not really here nor there. But what happens is when you shine light on this and you're turning from the trans to the cis, this peak gets smaller, this peak gets larger, because that's the peak that's due to absorbance from the cis molecule. When you're going from the cis to the trans, you see this peak get larger and that peak gets smaller. And so that's what we're going to be looking for when I show you these UV vis spectra in a second, okay? And so this is what we would expect to see. We would expect to see, in the absence of light, we have this giant trans peak. After we irradiate that trans peak, we'd expect to see it go down, and we would expect to see it progressively decrease as we hit it with more light, okay? What we've done is we've compared two different wavelengths of light, and it turns out that we get better isomerization with 366 nanometers than we do with 400 nanometers. That's neither here nor there, but we see that peak decrease. So when we tried this with our azo-resveratrol compounds, nothing. They do not isomerize. Actually, they do isomerize. This is a known effect. And this was buried in the literature, which I probably should have found before I set out to do these things. Because there's this resonance structure which causes these things. You can turn them into the cis, but they turn back to the trans so fast that you can't even measure it, right? And so we're like, okay, well, that was bad. And the reason is because they have this OH here, which is para to that NN double bond. And so we're gonna have to, if we're gonna pursue this research in the future, we're gonna have to make compounds that don't have an OH in that position. The problem is that the way to make these compounds is using an OH in that position. And so we had to develop new chemistry to do that, or we had to pioneer it in our lab. Okay, and so the next target we went after was this compound called cumbretistatin A4. Cumbretistatin A4 is from this South African bush. It was derived from so cumbretistatin is from the South African bush. They did all sorts of modifications to cumbretistatin, and they made cumbretistatin A4. This is a really good cancer drug, right? This and its sort of cousin, which is called ombrobulin. Okay. And so we're like, well, that has a double bond in it. Let's do the same thing, right? And not only this time, there's not an OH para to that double bond. And so we should be good when we're talking about the photokinetics. And so we designed this molecule. The idea was to put a nitrogen-nitrogen bond in here. We hit it with 400 nanometer light, turn it from the trans to the cis, and see if that worked. And so the way that this molecule works is that it interferes with polymerization of a protein called tubulin. So tubulin is what makes up microtubules. You may remember microtubules from cell division. They are what is responsible for mitosis. And so we see the microtubules shown here in green. Those microtubules pull the chromosomes apart during mitosis. And sort of like cumbretistatin, along with a series of other drugs, gets in between those subunits of tubulin, prevents them from binding. And so the cell division freezes. And if it freezes long enough, the cells undergo apoptosis. They're just like, something's messed up, and then they self-destruct, okay? It's actually what happens is they get caught in this spindle checkpoint during mitosis. And so that's good, because that kills cancer cells. We had to synthesize this molecule. The synthesis wasn't simple because, like I said, you couldn't just add two molecules together in a Sandmeyer reaction. So we took 1 and 2, we combined these in a Sandmeyer reaction. We made this molecule 3. We were able to methylate the para-OH group, right, in good yield to make this molecule. And then we were able to pull this allyl protecting group off in the last step using a little palladium catalyst to make our final molecule, okay? All right, so now we made this molecule, we got to do this photokinetic testing. And this is going to work, but there's a few things that we need to find. The first thing that we need to find is what wavelengths turn this from trans to cis, and what wavelengths turn it from cis to trans. And then it's like, how long do we have to hit it with that light in order to see the photoconversion that we want? And then once we've turned it from trans to cis, how long does it stay in cis before it just spontaneously goes back, right? And the reason we need to know that is because if we're going to test these things biologically, we need to know which one we're working with at any given time. Are we working with the trans, or are we working with the cis? Okay. And so this is our setup for these experiments. It's just a regular UV vis, and we have something really fancy. It's a flashlight we bought off Amazon for $7. Okay. So it turns out you can buy all sorts of different LED flashlights on Amazon, and we have them all. Okay. So this is what we saw, right? So we took our molecule, and we got kind of lazy because one of the things, nobody wants to stand there and hold a flashlight. Like the students would do it for a while, but at some point, even they were like, come on, this is ridiculous. And so we had to make a setup that would do this automatically. And so we used an Arduino, which we programmed to do this. We sort of parted out the flashlight, put it on a circuit board. And I can't take any credit for this. I had a student who was quite the engineer. She's now an anesthesiologist who did this. Anyway, and so we were able to shine light on this sample. This is with no light, 10 seconds, and we see progressively at about 60 seconds we've maxed out the amount of cis molecule that we can generate. Okay. So then we wanted to know like how long it would stay there, right? So we took the molecule, we irradiated it, right, with 400 nanometers, and then we just kind of watched it as it slowly turned from cis back to trans. And we see that after about an hour, it's about 50% reverted to trans, but after 7 hours, it still hasn't fully reverted to trans. And so what that means is that probably every hour, like if we want to treat cells with this for a long period of time, every hour we got to go in there, we got to like hit it with another burst of light for 1 minute. So when we did the cell tests on these things, we would hit it with 1 minute of light, and every hour we'd hit it with another minute of light. Okay. And then we were sort of curious, like, can we turn this thing back? Now, this was before we discovered that Amazon had like 470 nanometer flashlights and 530 nanometer flashlights. We're like, what turns it back? And so we took a white light flashlight, just a normal flashlight. And it turns out if you hit it with a normal flashlight, right, 30 seconds of white light, you can take the trans compound, you can put it in cis, you just hit it with a normal flashlight, you're already getting slightly back to trans, right? If you do 5 minutes with a white light flashlight, you can get all the way up there. You can revert this thing part way back. And the reason you can never get all the way back with this white light flashlight is because the white light flashlight has, of course, all of the wavelengths in it, including some 400 nanometer light. So you can never get all the way back. All right, so after we had done this photo testing, we're like, can we can we see if this actually works in a biological system? And so you can take tubulin, you can just buy the tubulin, right? And tubulin, if you just leave it together, it will spontaneously polymerize. And when tubulin polymerizes, it gets cloudy. And you can basically measure how cloudy the solution gets over time. And so we did 4 samples. We did DMSO. In the absence of anything, the tubulin polymerizes just fine. In the presence of combrettastatin A4, which is the model drug, right, nothing happens at all. And then in the presence of our drug in the dark, right, in the light, nothing happens at all. And in the dark, it polymerizes just fine. And so what we've demonstrated is that we can actually turn on our drug, which doesn't work at all, by hitting it with light. And then all of a sudden, tubulin won't polymerize. And that was kind of cool. That was sort of in a well, and that was with tubulin. And so we were like, well, does it work in cells? Can we kill cancer cells? And so we put some cancer cells in a well. We put our drug in the well. We incubated them while hitting them with light. And then we did a few tests to see if those cells were alive or dead. The first test is we just looked at them under a microscope, right? So if we take those cells and we just shine light on them, they don't die. Like light does not kill cancer cells. We've confirmed that. In the dark, they also don't die. These cancer cells are sort of like spread out. Okay, so each one of those is a cancer cell. If you take combrettastatin A4, it doesn't matter if it's the light or the dark, it's killing most of those cells. These round balls are dead floating cells, okay? But if you take our drug, right, in the dark, nothing happens. In the light, you kill almost all of the cells, okay? And so we can demonstrate that quantitatively. We can take those cells and then we can put a chemical in there. That chemical is called MTT. If the cells are alive, MTT is turned purple. If the cells are dead, it does not turn purple. And so we see that somewhere between 25 and 50 micromolar of the drug kills the cells in the dark, but in the light, it gets much more potent. It's somewhere between 100 nanomolar and 500 nanomolar of the drug. Okay, and we can graph those things, but it's really, we can graph those things, but it's sort of illustrative to sort of see qualitatively that change. Okay, all right. All right, so this is Ashton. Ashton was doing that work in 2013. She's now an anesthesiologist, okay? And this is us in 2015 when she presented her work at the American Society for Biochemistry and Molecular Biology. She actually won an award for that work, and we were so excited to publish it. And 6 weeks before we published it, somebody else published it in the journal Cell, right? Now, what I haven't shown, what I didn't say at ASPMB, I'm glad we're recording this, is that for 5 years I had been trying to get this work funded, right? I'd submitted 3 NSF career grants, and the reviews were like, this will never work, you can't make this work, whatever, right? And so I was really beside myself when they published this in Cell. And for those of you in the audience who don't know this, Cell is one of the best journals there is, right? And so not only does it work, people thought it was fantastic when somebody other than I did it, right? And so, not all was lost. We were able to publish ours just a few weeks later in the Journal of Organic Chemistry. Lots of people are interested in it. And it turns out we weren't the only people working on this at the same time. There was a group at Virginia Commonwealth University that published it 6 months later, right? So there were 3 people all had the same idea at exactly the same time. And I do want to say that the folks at Cell, Dirk Trauner and Oliver, we've gone to the first international symposium on photopharmacology together. They're awesome. And so I really have I can't hate. The good news is that it wasn't over there. So we were able to go back to the drawing board. We teamed with some folks at Texas State University in San Marcos. They made loads of derivatives. We made a couple. It turned gratifyingly, we made one little change. We turned a methyl group to an ethyl group, and we took the drug that they had patented and we made it better. And so we improved this drug, which was sort of gratifying. And we were able to publish that in the European Journal of Medicinal Chemistry. We're able to run these assays where you can sort of see the concentration of the drug over here on the x-axis and how many cells are alive on the y-axis. And you can compare the light and the dark, right? So we just kill tons of cells in the light. In the dark, it takes quite a lot of drug to kill them. And we can do 2 different types of cancer cells. These are, of course, the cervical cancer cells. I think those are liver cancer, but I don't know for sure. And we can then stain those cells. So this is the same experiment I showed you on the last slide, where live cells are yellow, dead cells are not. You see areas where there's lots of dead cells. And you only see those in the presence of light for our drug. Okay. All right. So that was fun, but at some point, you've kind of maxed out what you can do with azocombrettastatin. The good news is that there's this concept of a bioisoster, which is some molecule that does exactly the same thing as some sort of functional group that biologically kind of looks the same. And so azo groups are a bioisoster for loads of different groups: imines, amides, alkenes, alkanes, and ethers, all of which are found all over biology. And so there's like thousands and thousands and thousands of drugs that we could adapt this to. And so that's what we've done, right? So I have a giant research group, 24 students. And each one of them has a different target. We've made derivatives of inhibitors for a kinase called c-KIT, TIE2, PBP, which is penicillin binding protein, BCR-ABL, which I'm going to tell you about in a second, cyclooxygenase 2, and the PD-PDL1 checkpoint. Okay. But I first want, the one I want to talk to you about is chronic myeloid leukemia drug. So in chronic myeloid leukemia, this is what normal blood looks like. This is what you get in leukemia, where white blood cells are dividing so rapidly that they sort of outcompete and force out other types of blood cells. That happens because there's this chromosomal shift, right? So you have chromosome 9 and chromosome 22, and some people, they swap, basically. It's called a translocation mutation. And so this bit of chromosome 22 comes over here to chromosome 9, and this little bit of chromosome 22 ends up with a little bit of chromosome 9. And then you get this small chromosome, which they refer to as the Philadelphia chromosome because it was discovered in Philadelphia at the University of Pennsylvania. Okay? And so what it does is it takes two genes, BCR and ABL, and it fuses them together. And that creates a kinase, ABL, which doesn't shut off ever. And because that ABL kinase is responsible for cell division, you get cancer. Okay? And so this is kind of what it looks like. There's all these like different intermediates. There's a lot going on here, right? But there's different intermediates, and each one of these things leads to cell survival or cell progression, cell development, or mutagenesis. And basically up here, right, at the tyrosine kinase level is ABL, and so it controls all of that. All of that stuff gets turned on when when you've got the BCR-ABL mutation. And so people were excited. Man, people were excited. In 2004, which is when I graduated college, this was on the cover of Time magazine, okay? There are not very many drugs that make the cover of Time magazine. And this one was Gleevec, it's brand name Gleevec, its generic name imatinib, which was a selective BCR-ABL inhibitor. And so it was an inhibitor that was able to inhibit BCR-ABL without inhibiting too many of the other 550 kinases that are present in our bodies. And so I was like, well, it turns out that so since the imatinib, right, what happens with imatinib is that it causes cancer to go away for a while, and then eventually BCR mutates, so the imatinib no longer binds. We made varieties of generations of these drugs: dasatinib, bosutinib, nilotinib, ponatinib, asciminib, are all successive generations of the drug which fight different mutants of BCR-ABL. And so I can't tell you about the synthesis or the exact molecule that we've made, but I can show you some of the results, okay? Because we're trying to patent and publish that at the moment. So the drug that we made, right, we inserted an azo bond into a BCR-ABL inhibitor. We're able to show, this is hot off the presses, we were able to show that we can turn that drug from the trans to the cis conformation, and it takes about 5 minutes to do that fully. We're able to turn it back from the cis to the trans conformation, and it takes about 30 seconds to do that. So we start here in the cis conformation, we hit it with 470 nanometer light, and we're able to turn it back into the trans conformation. And then we were able to test that drug in vitro using an assay. So we put BCR-ABL into a well, and we see if we can inhibit it, right? What we see, so this is on the x-axis here is the concentration of drug, and on the y-axis is how active the enzyme is, okay? And so we see that as the drug concentration increases, the activity of the enzyme decreases, and it does that at a much greater concentration in the trans azo log, and it does that at a much lower concentration in the cis azo log. And so we see about a 12-fold increase in activity when you shine light on this thing, and it's a sub-micromolar drug, which is decent. So sometimes people ask, right, and they say, okay, so chronic myeloid leukemia is a blood cancer. What good is it to target it, right? Well, you want to inhibit the BCR-ABL without messing up other kinases. While that blood is everywhere, it is possible. This is just an engineering challenge, right? And so you have blood vessels which have BCR-ABL in them. Actually, that's not totally true. You have a drug molecule. You are able to activate the drug molecule. The active drug molecules will bind to the BCR-ABL, right, inhibiting the BCR-ABL. The inactive, the active molecules can then be inactivated by an inactivating light before they spread to the rest of the body. And so we actually think that this is a really promising way to sort of target a cancer that would otherwise be untargetable, or not easily targetable. All right, how much time? Are we done? 15 minutes, perfect, because I want to tell you about Paul and Southwick versus everyone, okay? So Paul and Southwick are two of my research students, and it's cool. So every drug I've shown you so far is like we took a carbon-carbon double bond and we replaced it with a nitrogen double bond, and that's hard because the syntheses are hard. Sometimes these syntheses are 10 steps, right? It takes people, I mean, the synthesis of the BCR-ABL inhibitor I just showed you took Noah, who's in my research lab, it took him like 3.5 years to complete, right? He started with me his second semester, and he just finished it like probably 2 months ago. And so it's like, could we do this in a more efficient way? And the pharmaceutical company already does this. They use what's called parallel synthesis. So they'll take two molecules and they'll mix them together and they'll do the same reaction like a gazillion times, okay? And so they take A plus B, A plus C, A plus D, A plus E, and each one of those is making a different molecule, although they still have the same A core in them, but they can do the same reaction over and over and over. And the reason that's really good is because robots are really good at doing the same thing over and over. Now, so really I was trying to turn Paul and Southwick into robots, okay? And so the question was, instead of putting an azo in like the middle of a molecule that required us to like sort of assemble the whole molecule in like many, many steps, could I take like drug molecules that somebody else had made, and could I just slap an azo on it and make a drug that would turn on and off? Now, a lot of times when you put an azo on a drug, it's not going to work, but if I could do it on thousands of drugs in a single step, that would be really cool. And so the first thing we decided to go after was antibiotics. And the reason I wanted to go after antibiotics is because nobody else is. And so if you want to do a thing that's competing with the pharmaceutical industry, maybe you should find an area they're not in. And so the pharmaceutical industry has almost entirely divested themselves from antibiotic development. So we see a drastic rise in drug-resistant bacteria, but I think it's every pharmaceutical company in the United States but one has divested themselves from research. And so we know that 10 years from now, the problem is going to get worse, okay? And so there's all these cool things about bacteria too, right? It's like humans have 30 trillion cells, but inside of us there are 100 trillion microbial cells, right? And so somewhere between 70 and 90% of all of the cells inside of us are not us, they are bacteria. And what gets even crazier is that the genes inside of us, 99% of the genes, that is to say 99% of the proteins that get made inside of us are not our human genome. 99% of it is the microbial genome, the microbiome, okay? And these bacteria, like some of them are bad, we think of bacteria as bad, but many of them, those tend to be good, right? They do all sorts of things. They help our immune system mature and function. They help our brain develop. They help protection and clearance from pathogen invasion. They help do metabolic things. And they provide all sorts of nutrients, thiamine, folate, niacin, biotin, okay, for us, many of which we can't produce ourselves. And so you can imagine that it would be really useful to kill the bad bacteria while keeping the good bacteria. One of those bad bacteria is C. diff, right? Clostridium difficile, right? C. diff is one of those infections where you kill all the good bacteria. It's sort of opportunistic. It sort of takes over. You see there are like 250,000 cases in hospital patients, and this was in 2017, the last year for which I could find CDC records. There's an estimated 13,000 deaths, and it cost the healthcare system over $1 billion. And that was pre-inflation, by the way, so $2 billion now. All right, so the way that penicillin works, right, is that and we learned this in biochemistry, is that your bacterial cells are making peptidoglycan cell walls. Those cell walls have a bunch of sugars in a row, and they make these like long lines that get stitched together by amino acids. Putting the sugars together is called transglycosylation, and then stitching those strings of sugars together is called transpeptidation. Penicillin primarily works by inhibiting the enzyme that does transpeptidation. And it does that because it has this weird beta-lactam structure in it, which mimics the D-ala amino acids that are set up in peptidoglycans. So sometimes people ask, like, what is the natural stereochemistry of amino acids? I'm like, in bacteria, there's a lot of D amino acids. Okay. So the question is, we would like to just slap an azo group on this thing, and one way to do that is making ester bonds. We learned how to make ester bonds in undergraduate organic chemistry, and so that we could change the structure of this thing and hopefully turn it on or off. We don't know which one will be on or which one will be off, but we'll figure that out later, details, right? And so we modeled it, right? We took penicillin binding protein, which is where these things bind. We modeled where penicillin binds. There's not a really good model for this because this reacts with penicillin. And what reacts with penicillin is that green residue. And because the green residue is right where we would expect it to react, which is at that beta-lactam ring, we know that our modeling looks pretty good. This is freeware modeling, by the way, UCSF Chimera and Autodoc Vina. So like, because free is good for us, right? All right, so we took the drugs that we wanted to make, we put an azo group on them, and then we docked them in penicillin binding protein. Then we gave them scores because Autodoc Vina gives them scores. Those scores are not very far apart, which suggests that both the cis and the trans are kind of going to bind to this thing, which means that we're not going to be able to turn the drug on and off. But I don't care. We're going to try to make it anyway and see what happens, right? And so there's a synthetic challenge because the way that you tend to make esters or amides is that you activate this carbonyl and then you attack that carbonyl. If you put anything in there at all that is a good nucleophile, you break that beta-lactam ring. And so you have to do this in a very different way, right? We actually had to, instead of using a nucleophile to attack this carboxylate, we had to use the carboxylate as the nucleophile to attack an electrophile. And so what is a really good electrophile? A benzyl tosylate. It's a leading group from organic chemistry. And so we're able to make this benzyl tosylate, but it is such a good leaving group that water in the air reacts with it. And so here's Paul versus everybody. He's in the glove box at Albion. He has to make this molecule in the absence of solvent. So he puts all the reagents in. It's the weirdest reaction I've ever seen. He puts all the reagents in, he grinds them up with a mortar and pestle, and then at the end, he's got his product in like, 37% yield, right, which is pretty wild. Then he mixes that with penicillin. This yield is now 30% yield as well. And he makes this final compound. Now, there's a lot going on here, and I don't worry about A, B, D, F, or A, B, or G, okay? I want to draw your attention to A, D, and F. So these are just, these are what are called Kirby-Bauer assays. You can literally put this drugs on a little piece of paper, and then you can put it on bacteria, and you can see like how far the bacteria are killed around this antibiotic. There's a DMSO control on this plate. This is penicillin with this bacteria, so there's like these big zones of inhibition. You could measure those to basically tell how good your antibiotic is. And then we did this with our drug. Now, the zones of inhibition are not nearly as large as they are with penicillin G, so we have reduced the activity of this drug, but we're willing to reduce the activity of our drug if we can increase our ability to target it. The problem is that the zones of inhibition are almost identical in size between the drug that was sort of exposed to the light and the dark, and that doesn't really surprise us because when we did the modeling showed that they would be kind of the same. And so we went back to the drawing board, right? Can we make derivatives that will have giant differences between the binding of their trans and their cis? And so the carboxylic acid didn't, and the 4-phenyl group didn't, and the 4-methoxy didn't, and the 4-fluoro didn't. But this one did. And so what we did is we went back and we made that compound, okay? And I don't have the results here today, but it works, right? So we just found out like 3 days ago that we see dramatic differences at 100 micromolar between the light and the dark for this drug, which is pretty cool. So, all right. So that was most of what we do in my lab. But there are other ways that we think about these things, and I've got, sounds like 5 minutes left. So okay. So now, if I've got 5 minutes left, I will tell you about the other thing we're doing with antibiotics. So there is the antibiotic apocalypse by 2050. We expect to see 10 million deaths per year. I mean, it's hard to even like wrap your head around that. That's coming from antibiotic-resistant infections. Today, there are 700,000 worldwide. So it's a big deal. Bacteria become resistant in a variety of ways, right? In a large population, a few are just resistant genetically because bacteria have all sorts of genetic variants. So you kill off the ones that are not resistant, the resistant ones then grow back, and so now all of them are resistant. And what's even more pernicious about these things is that they can take those genes and they can pass them to other bacteria. So, you know, we use, you know, soil bacteria become resistant, and then they can pass it to bacteria that colonize, they can pass those genes to humans, which, you know, put people in the hospital, for example. And so what we were inspired by in our sort of this other project that we're doing is this of glycopeptides. So there's this antibiotic which is called vancomycin. Vancomycin was long called the antibiotic of last resort because when nothing else worked, you could give people vancomycin, would take care of the infection. But it was last resort because it had some pretty nasty side effects. And the way that it worked is it would take these, so these are the same, this is the same peptidoglycan I showed you a few slides ago. It would take this amino acid tail on peptidoglycan, it would cap it. And when it was capped, transpeptidation couldn't happen, okay? And so what we want to do is we wanted to create antibodies that would cap these things. And the reason we wanted to create antibodies that would cap these things is because antibodies you can evolve, and so we could fight evolution with evolution. And so the way that this works, now we're going to take full antibodies because full antibodies are like expensive. What we're going to take are what are called nanobodies. So we've taken an antibody which has 12 domains in it, and we cut this one off. And this is sort of the business end of the antibody, which is a nanobody. And it has 3 regions, 3 loops of amino acids in this protein, which basically grab things. Okay, and we're hoping, those are called complementary determined regions. There's 3 of them shown here. We're hoping to evolve those things in a way that allows us to cap this, just like vancomycin. And so what we do is we take yeast. And those yeast, somebody has already done this. I don't have to do this. You could buy this commercially. Somebody has taken yeast, a yeast library with billions of different yeast. Each yeast is displaying a different nanobody on its surface. And they're just kind of random, right? But you can take those yeast that each have a different nanobody, and you can put them in a well that contains a bead with the nanobody on it. And then those yeast that have a nanobody that bind to your antigen, the thing that you want to create a binder for, those stick, right? All the other ones get washed away. We're able to like hold on to the beads, right? And we do that using magnetic activated cell sorting. It's magnetic activated because those beads are magnetic. They literally stick to a magnet. We wash all the other yeast away. Then we take the magnet, we depolarize it, and the binding ones pop into some sugar water, because that's what yeast need to grow. Then we grow up a whole bunch more of them, and we repeat the process. So you might start with 3 billion pairs. After the first round of selection, you might be with 100 million pair, maybe 10 million different yeast. And then the third selection, you have 1 million yeast. And the fourth, you're down to 10,000. And you do about 6 rounds of selection, and you can get 150 different yeast. Okay? And this just shows kind of how that happens. This is our magnet, right, over here, how we're doing that selection. Okay? And I wish I had, so this is really kind of an exciting project. And what is exciting about it is choosing the antigen. And so we have developed the antigen that is necessary to do this selection. And so we're in the process of doing multiple rounds of selection to make nanobodies that bind to basically the lipid that makes peptidoglycan. Okay. All right. Now, I'll skip the last bit of how we're using computers to do this stuff. I was being a little ambitious. But I would like to thank all my students. So I have like a giant lab. You know, this research happens so slow. This is, 8 years of research probably through 25 or 30 different students. And we do lots of fun stuff. You should do research because, the research students study together. They share materials. Like the juniors help the sophomores study together. We were able to do all sorts of fun things, throw axes, work in the lab. Down here we got to go to Tulane and see things. We got to go to Times Square and the Liberty Bell for various conferences and whatnot. So that's it. Thank you very much. Any questions?
Okay, so I just have like 3 questions. So the first one, you said the translate to the cells.
Yeah.
You talk about like a leukemia cell, like hemophily in the blood.
Yeah.
Does the photon also translate to skin also going to?
Yeah, so that one's kind of interesting because the active form of the drug is actually the trans form of the drug. So we can hit it with 470 nanometer light, which does penetrate relatively far. Yep.
Also, does the translate like the metabolism is a little bit different because you change the structure, so maybe pharmacokinetic from that.
Oh, absolutely. I'm sure it changes, right? Yeah. So we still have to do all that testing. So all that we've really done is if we've gotten rid of the first 2 or 3 years where you're trying to find a bioactive substance. We start with bioactive substances, but we don't know. And in fact, these things do get metabolized. They get metabolized differently, right? So like when you after you eat a candy cane, it doesn't come out red. You know what I'm saying? So they do get metabolized, right? But there are some that don't, right? There's a UTI drug called azo, which you eat it's orange, it comes out orange. And so changing the electronics around these things does change their pharmacokinetics a lot, right? Yeah.
I think my third one. Oh, yeah, my third one is that because you use the UV light to treat the bacteria, but also you have to also do the negative control for like UV. Maybe some people can argue maybe UV light directly also kills bacteria.
Absolutely. Yeah, so we've done those controls. And that was, I was like, don't pay attention to these other ones, but that's what they were. They were light by themselves. But that's a really good point, yeah. But yeah, they were up there. I just didn't focus on them, yeah, yep.
Any other questions?
How do you produce the nanobodies? Like, do you make them individually?
Yeah, so the yeast have a gene inside them for the nanobodies, and I was sort of running short on time, but the nanobody is fused to a protein called AGA. So yeast naturally express this AGA protein, which always goes to their surface. And so when you fuse that AGA protein with the gene for the antibody, it automatically goes to the surface. And so these things are commercially available. In fact, something like 50% of the drugs that you take today that are commercial, that come out on the market each year are now biologics, not small molecules. And they're produced in one of 2 ways. They're either like you give an antigen to an animal and you isolate the antibody out, or you do selection in this way. And so there are companies that specialize in this. And you can sort of like buy their commercial versions of this, yeah. Thank you so much.